Francis Crick developed the adapter hypothesis to explain the function of tRNA in the cell. When I thought of the word adapter, I immediately thought about old headphone adapters that Apple released when they changed the shape of their charging port. In this illustration, the mRNA is represented by the phone. It holds the information directly transcribed from the DNA. The tRNA is represented by the short, white headphone adapter cord here. The tRNA has one end that is able to pair with the mRNA, and one end that picks up the amino acid. The amino acid is represented by the headphones. In a cell, the mRNA attaches with the various components that make up the ribosome. The tRNA is then able to act as the adapter by being able to connect the transcribed DNA code that is present on the mRNA in the form of a codon with the anticodon that translates the DNA's information into a language that the cell can read and use to form a protein. In the same way, the headphone adapter here is able to interact with the specific charging port on the phone on one side and the headphones, or the amino acid, on the other side. Without the help of the adapter to translate between the phone and the headphones, the two could not interact. Just like how tRNA enables the cell to use information on mRNA to create proteins. An alteration that might prevent tRNA from following the genetic code would be a change impacting the ability of the tRNA anticodon and the mRNA codon to wobble base pair. For example, if the tRNA had the structure of the guanine altered so that it could only bind with C instead of both C and U. In a normal cell, if the mRNA base pair read CAU 5' to 3', then the anticodon it could base pair with could read GUG. However, with the hypothetical alteration that would prevent guanine and the tRNA from being able to base pair with uracil, this pairing could no longer occur. The only tRNA anticodon that would be able to pair with the CAU codon would be the GUA anticodon. A change in tRNA's ability to wobble base pair would reduce the redundancy of the genetic code. A single tRNA would not be able to bind with as many mRNA codons as in a normal cell. Some tRNAs would have to work overtime to compensate for pairs that can no longer occur. Proteins would be produced with some missing or changed amino acids. This could change or totally alter the function of proteins within the cell.